Thanks, Angel. Our next speaker is Professor Andrew Owen from the University of Liverpool. Andrew works at the interface of nanomedicine and infectious diseases with focus on HIV. And I met him 2 years ago in Clinam. And he's also undertaking his developments to the market through a company that was founded at the university. And I took HIV as a model of infectious disease because it's probably a paradigm of infectious disease and where we are seeing some developments at the interface of drug delivery systems and nanomedicines. So Andrew, please.
Thanks very much, Andrew, and thank you for organising the session and inviting me to speak. So I'll start off giving some of the background to HIV therapy and some of the current challenges which exist within the HIV area, and particularly those which are relevant in the context of the paediatric considerations. So the 2014 WHO statistics estimated that there are 2.6 million children under the age of 15 years which are HIV positive. And quite worryingly, only 32% of infected children are currently on therapy. More broadly speaking for HIV, demand for some of the preferred drugs within resource-limited settings is outstripping current manufacturing capacity, meaning that we, as a species, cannot make enough of these drugs to treat all the patients which are currently infected by HIV. So it follows then that any strategy that reduces the doses required for effective viral suppression will increase the number of patients able to receive therapy, and depending on the technology being applied, that may also have the potential to reduce the costs of therapies, and that's particularly important in the resource-limited context. New formulations for children, as Alejandro already said, are urgently required for HIV, and I'm going to talk a little bit about that within the presentation. There's also other limitations of current APIs for HIV, and these are summarized here. Alejandro already touched on the patient adherence issue, and I'll talk about that in more detail in my presentation. I think that HIV and infectious diseases per se, but particularly HIV adherence is particularly important because patients have to adhere to at least daily oral intake of their medication, because it's a chronic disease that's a lifelong commitment to their therapy. Of course, with infectious diseases, there's also the potential for emergence of resistance, and so adherence is even more important in the context of infectious disease. Many of the drugs used in HIV have a very poor oral bioavailability. There's a huge inter-individual variability in the pharmacokinetics, and this is partially explained by our current knowledge of the pharmacogenetics of these drugs. Some drugs exhibit fed-fasted variation, and adverse drug reactions are an ongoing issue, as they are in many other diseases. So one of the things that so I'm going to split my presentation into two pieces. One is some of the work that we've been doing in Liverpool on development of oral formulations, which we think are particularly relevant in the context of pediatrics. And the second part of my talk is going to focus on the new long-acting nanomedicines, which are starting to emerge from various large pharmaceutical companies, and some of the issues around those formulations in regards to pediatrics. So starting with the Liverpool oral nanomedicine development program, we've been working for around 7 years now using an approach which was published in Nature Nanotechnology back in 2008. It's actually quite a simple approach. It starts with a emulsion of an organic phase and an aqueous phase, with the drug dissolved within the organic phase of that emulsion. You can subsequently freeze-dry that emulsion. As a result of that freeze-drying process, you form a dry, porous structure with zones of the organic compound or nanoparticles of the organic compound or the drug within that porous structure. It looks a little bit like this under an SEM. And then subsequently, when you disperse that dry, porous structure back into an aqueous environment, the porous excipients dissolve into the water, but the insoluble drug is released as nanoparticles into a nanodispersion. To achieve scale, you can also conduct the same manufacturing process, but using a spray drying approach rather than a freeze drying approach. And we've been working across library generation by freeze drying, and then achieving scale for clinical batches using spray drying manufacture. So nanomedicine, I think, is multidisciplinary. There's an unprecedented need for interdisciplinarity in nanomedicine. We're quite fortunate in Liverpool to have a very strong pharmacology and chemistry link. And the strategy that we've been employing for this oral nanomedicine development program is excipient screening. So typically we can generate a library of solid drug nanoparticles containing around 200 different formulations with different sizes, different excipients, different zeta potentials. And we could do that within a couple of weeks. And then physically characterize those libraries before moving them into an advanced program of pharmacology-based lead selection. So typically we're doing in vitro selection, in silico PK prediction, followed by in vivo confirmation. And once we have identified using a freeze-dry library formulations with a optimal in vivo performance, we can then transfer the manufacture to a spray dry manufacture and go through GMP translation. Oops. So where we are at the moment is we have 2 antiretroviral formulations with in vivo confirmation that we can achieve a 50% dose reduction while maintaining the pharmacokinetic exposure to those drugs. We've been through regulatory engagement and approval, and those formulations are currently in clinical validation in healthy volunteers. Now, this is why we think that these formulations are particularly interested in the context of pediatrics. This is one of the drugs we've been working on. It's a drug called lopinavir. It's given as a combination with ritonavir in a brand called Calitra. Now, this is the current pediatric formulation of lopinavir. You can see down here it contains 42.4% alcohol by volume, and 15.3% propylene glycol by weight, by volume. So this is effectively giving lopinavir to very young children in a shot of vodka. And there's clearly issues relating to that, and this is one of the reasons why new formulations for children are urgently required. So this isn't lopinavir, but this is a dye which has been processed using the Liverpool solid drug nanoparticle approach. The bottom has been processed into nanoparticles. The top is just an insoluble dye. It does what you expect. It doesn't wet. It doesn't dissolve. At the bottom, after processing into solid drug nanoparticles and dropping into water, you can already start to see it streaming down into the water. And when you give it a shake, at least by eye, it's physically indistinguishable from if it were in solution. So there's a clear no-brainer there on whether you want to give formulations to children in high alcohol content solutions or as a nanoparticle dispersion in water. So this is where we are at the moment. We've conducted lead selection. We've translated a CGMP. We've got long-term stability on powder-filled capsules, as well as in the liquid phase. We've got regulatory approval, and the first patient or healthy volunteer, I should say, enrolled in December 2015. We've got some very encouraging data coming from this clinical evaluation, but I can't, unfortunately, talk about that today. So I want to get back now to the long-acting formulations which are emerging, and I want to get back to adherence. This is a pharmacokinetic profile. I'm sure everybody's seen them before. This is for a once-daily oral regimen for a drug. So this is classically what the pharmacokinetics of an antiretroviral drug would look like. You've got the plasma concentration on the y-axis and the time in days across the x-axis. And you see a patient takes an oral tablet, you get a peak, trough, all the way up to steady state. And then the goal of therapy is to maintain the pharmacokinetics within this therapeutic range. Unfortunately, patients aren't adherent to therapy, and this is just a small study here. I'll just talk you through this. Each one of these bars represents a different patient. Where it's green, the patient is adherent to their therapy. Where it's red, the patient is classed as non-adherent, either they've missed a dose or they've missed several doses of drug. You can see this is a science in itself. There's some patients which are very non-adherent, some patients which are perfectly adherent. So there's a whole science around adherence anyway. But what impact does adherence have on the pharmacokinetics? Well, you can see if a patient misses just 3 doses of their drug over a month, you dip down below the therapeutic threshold for this drug, and the virus has the potential to escape the drug through formation of resistance mutations. But if a patient misses more doses of their drug, then there's a cumulative effect because the drug dips down and needs to go back up to steady state. So this has been a major driver for the long-acting formulations which are now starting to emerge in HIV, where the fundamental principle is you give 1 dose of the drug on day 0, and you get therapeutic exposure over an entire 1-month interval. Now, this is particularly important in terms of pediatrics as well. We know that adherence is a particular issue, particularly during adolescence. This is one study which was published in AIDS back in 2014. And just to summarize this, the overall adherence, percentage adherence within this study was 62.3%. So only 62.3% adherence in adolescents to their antiretroviral therapy. So clearly there's a lot of talk now about validating these long-acting nanomedicines for use in this particular subgroup of patients. There are currently 2 which are in late-stage clinical development. The first was developed by Janssen. It's a rilpivirine LA formulation and comfortably gives 1-month exposure to the drug from a single intramuscular depot. The other one is cabotegravir, developed by GSK and being marketed through ViiV Healthcare. And this drug comfortably gives 2 to 3 months exposure from a single intramuscular depot. So clear excitement in the field, potential for application and treatment as well as pre-exposure prophylaxis. Potential problem is that there's only 2 drugs currently in development, and most antiretroviral drugs are given at least 3 drugs. But there are currently many unanswered questions regarding the mechanisms of drug release from this nanomedicine within the local depot. And I'm just going to talk through some of the question marks around this. This is a cartoon depicts the muscle cells with the blood capillaries over the top and the lymph capillaries with the depot administered within that intramuscular depot and the nanoparticles dissolving into drug within the depot. If we look at the structure of some of these capillaries, we know we have a tight endothelium around these capillaries. The intercellular clefts are probably too small to allow nanoparticles to enter directly into the blood capillaries. But these cells, are metabolically active. They express transporters, they express cytochrome P450 enzymes, and there's a real need to understand the role that these metabolic processes play. Similarly, in lymph, now we've got intercellular clefts which probably are big enough to let nanoparticles directly into lymph, and that's been demonstrated in preclinical species. And again, we've got metabolically active cells around this capillary. But as Alejandro described, we know that there's differences in metabolism and in the gastrointestinal development in children throughout childhood. So what about these processes? How are these processes going to be different between children and adults? I think there's a lot of unanswered questions there. What we do know is you get macrophage infiltration into the depot, which phagocytoses the nanoparticles, and very quickly after administration of the depot, those macrophages form a granuloma, which may constitute another barrier to drug release into the systemic circulation. So a lot of questions regarding what regulates drug release, and even more questions for the differences between adults and children. So I'm going to leave it there. I want to flag up that the NIH have funded a long-acting and extended release resource program that we're part of. It's being coordinated out of Johns Hopkins University in Baltimore. So if you're interested in the long-acting side of this, then I would encourage you to go and take a look at the resources that we're collecting and putting on here. A shameful advert for our new journal of the British Society for Nanomedicine, which is a journal of interdisciplinary nanomedicine. And then I'll just leave you with some acknowledgements, particularly Steve Ranod, who's the materials chemist that we're working with, Marco Siccardi, who does all the PBPK modelling, and Neil Liptrot, who's in the audience, who's leading the Liverpool contribution to the European Nanotechnology Characterization Laboratory. Thank you.